Hi guys, hi guys, we are going to go through proteins. So this is specification again. I'm going to be showing you this at the start of each and every video, and do have this in front of you, and I'll be referring to this as well. So what are proteins? Now, monomers of proteins are amino acids, and you should know what monomer is. It's basically the building blocks of a polymer, suggesting that these proteins are a form of polymers. And so a chain of amino acids, you do have to know what they look like, but for now let's represent them as squares. So a chain of amino acids can make what's called a protein. Now, a dipeptide, okay, I'm just hitting random words at you now. A dipeptide is formed when two amino acids, so amino acid 1 plus amino acid 2, so when two amino acids join together via condensation reaction, you get a dipeptide. So two amino acids joined together forms a dipeptide. A polypeptide is when a chain of more than two amino acids are joined together, which gives a polypeptide, and this polypeptide is referred to as a protein. So this suffix here, peptide, monopeptide, well, monopeptide is an amino acid, but peptide or dipeptide or polypeptide or tripeptide, the word peptide always refers to proteins. And whenever you think of proteins, think amino acids. I'll be abbreviating amino acids to AA, just because, yeah, I can't be bothered to write the whole word. And a protein is one or more polypeptide. So here is what an amino acid looks like, and yes, you do need to know what an amino acid looks like, and you need to be able to draw this in the exam, and it's also mentioned on the spec. So there are 20 different amino acids found in almost every living organism. Well, for us A-level students, GCSEs, etc., you've just got to know that there are 20, but there's obviously more than 20, but for now stick to 20. So all of these 20 different amino acids have the same general structure. So we've got what's called our amine group, we have a hydrogen, and we have what's called an acid or a carboxyl group, and we also have what's called the R group. So you probably came across the R group during lipids when you were revising or going over lipids. So the amine group, which is NH2, you've got a hydrogen, and you've got a carboxylic acid or a carboxyl group, and then you've got the R group. R group can be anything, literally anything. You could have lots of different, usually we're just substituting carbon, the R, for anything which is like really long or complex, etc. And this R group is what differs between each amino acid. Otherwise, every amino acid must have an amine group, a hydrogen, and a carboxyl group. So these are just examples of different types of amino acids. So we've got amine, hydrogen, carboxyl, and then we've got our R group here, and our R group here contains hydrogen, and glycine is the only amino acid which contains which has an R group which has a hydrogen in it. Otherwise every other R group would have a carbon in it. So okay, I think I messed this bit up. So R is down here. So we've got our R group, we've got our carboxyl group, we've got our amine group, and we've got our hydrogen. Here the R group is a methyl group. You don't need to know that, but it is. And so it's got a carbon with 3 hydrogens, and then again we've got our amine, hydrogen, carboxyl group, and then we've got our R group. R group this time contains a carbon, 2 hydrogens, 1 oxygen, 1 hydrogen. Then again amine, R group and carboxyl group, and our hydrogen. So yeah, get used to it. Okay, so a condensation reaction between two amino acids forms a peptide bond. A peptide, always think proteins. Every time you see the word peptide, think amino acids. So dipeptides are formed by the condensation of two amino acids. So I mentioned this before. So you've got two amino acids, join them together via condensation, which releases a water molecule obviously, and you form a dipeptide. Now here's a better example, and you could, and it has come up in the past before, where you have two amino acids, draw them together, and you have to form your dipeptide. And you also have to know what is the peptide bond. So the peptide bond, obviously, when the oxygen and the hydrogen on one amino acid and the hydrogen on the other amino acid are lost, the thing that is joining these two amino acids together is this bond here. So this bond attaches to the nitrogen here, and this whole section is called the peptide group, and that is the peptide bond. Now the role of hydrogen bonds, ionic bonds, and disulfide bridges in the structure of proteins, blah blah blah, I'm going to go through this. So let's look at the different protein structures. So these protein structures essentially describe how they develop over time. So what I mean by that, so we've got our normal amino acid, add another amino acids, we get a dipeptide, yep, and then if we add like 400 more of these amino acids on, we get what's called a polypeptide, just a simple basic chain of amino acids. This is your primary structure. And so don't worry, I'll go through all of these. So understanding protein structure is crucial, as each protein has a specific shape. So understanding this will help us understand any abnormal folding of the proteins and where things have gone wrong, etc. So the primary structure of proteins. So this is a sequence of amino acids, or a chain sequence of amino acids in a polypeptide chain. Literally this, you've got a chain of amino acids and that's it. That is literally it. So you've got thousands of the, well, I say thousands, or however many there are of these. So usually, like, let's say 3 or more. So here's a little question. So I've got my amino acid chain here. It's a primary structure, so I'm going to abbreviate that as 1. So on this end I've got NHH, which is an amine group. So that's at the end of this amino acid here. Can you take a guess at what would be at this end here? So if amine is here, what would be at this end? Well, obviously it's going to be your carboxyl group, and that's always going to be the case. So even with our other amino acid diagram, amine's always at one end and the carboxyl group is always at the other end. Okay, so now let's look at the secondary structure. Now we're going to involve these types of bonds. So the polypeptide chain sometimes coils. So we get our normal primary sequence chain and primary structure, and suddenly it decides to coil, and it produces either a spiral, so here, or like a pleated type sheet. Now this spiral is called an alpha helix, and this pleated sheet is called a beta pleated sheet. Now this occurs when two or more parts of the chain run parallel to each other and are linked to each other by hydrogen bonds. So in a beta pleated sheet, you've got when two parts of an amino so let me try and draw this out. So you've got your polypeptide here. I know, very good drawing. So what's happening here is they're like running parallel to each other, and suddenly what's going to happen is they're going to form hydrogen bonds, and that's going to like create this beta pleated sheet right here. And alpha helix, like, it just occurs, it just happens because of the formation of these hydrogen bonds. Because basically when you like squish all these like amino acids together, which are in a chain, they can either form a beta pleated sheet or an alpha helix, depending on like their position, etc. Looking at the tertiary structure, carrying on from the secondary structure, the polypeptide chain is folded even further, so it's going from a chain sequence of amino acids, then it's coiled into either an alpha helix or a beta pleated sheet, and then it's folded even further, and therefore, because it's like kind of like scrunched up all together, it's going to obviously form more bonds, and you get an extra two types of bonds. So you get ionic bonds and disulfide bridges. So before, in our secondary structure, we only had two types of bonds going on. So we've got peptide, obviously, which is between the amino acids, and now we've got hydrogen bonds, which are here. So here we're gonna have ionic bonds and disulfide bridges. So here, hydrogen bond, and here's disulfide bridges, which is between, specifically, you don't need to know this, two cysteine amino acids. And these cysteine amino acids contain an element called sulfur, and sulfur forms like covalent bonds between another sulfur, and yeah, that creates a disulfide bridge. And then you've got ionic bonds, and that's to do with opposite charges on different amino acids, and yeah. So you have two categories of proteins differing in tertiary structure. So you have a fibrous protein, so that they're like long and thin, and they are insoluble. They have a structural function, such as keratin in hair or collagen that makes up a lot of connective tissue in our bodies. Obviously, you don't really need to know this, but it's a good idea to just have a basic idea of what these two different types of proteins are. Then you also have globular proteins, which are more spherical in shape, and they are soluble and have a biochemical function such as enzymes or myoglobin, a pigment which stores oxygen in muscle tissue. So these are like the general differences. If you want to memorize it, you can, because it could be beneficial for your essay in paper 3. But you've got a long time for that, or unless you're in year 2 and you're, yeah. And the last final stage of a protein structure is the quaternary structure. So this is where two or more polypeptide chains has like the same interactions and stuff, and yes, literally two tertiary structure proteins just come together. That's it, and you get your own very own quaternary structure. Hemoglobin is an example. So it has 4 polypeptides. 1, 2, 3, 4. Yeah, you can tell by the color. So to test for proteins, what we're going to do, we're going to do this thing called a biuret test. So we're going to get copper 2 sulfate plus sodium hydroxide. That makes a biuret solution. Now usually I prefer to stick to biuret solution. So I'll say, oh, I get biuret solution, then add this biuret solution to the protein solution, then observe to see a color change from blue to purple or mauve or mauve, or however you're meant to pronounce that. They could in the exam say, oh, Jonathan mixes copper 2 sulfate and sodium hydroxide, then he adds it to a test tube and he sees a color change from blue to mauve. How do you know what's in this like solution? Well, obviously it's going to be a protein, because this copper 2 sulfate plus sodium hydroxide is what makes up the biuret solution. Remember, enzymes are proteins as well, and we'll cover enzymes next.